Well, welcome everyone. My name is Chad Jackson. I'm the Senior Director of the Preclinical Translational Research Program at the Foundation Fighting Blindness. From the notification here, I see people are still joining the meeting, so we'll give it just maybe 30 more seconds for people to join and then we'll start. All right, I had a certain goal of participants in mind and we've hit that number. So I think we're okay to get this webinar started. So first and foremost, I'd like to thank everybody for taking time out of your day to join the Foundation Fighting Blindness for one of our educational webinar series. We're really gonna have a very robust discussion today with our teacher today. And I'm so happy for her to be here. Dr. Cheryl Rowe-Rendleman is going to take us through some of the how to navigate the FDA and EMA, and especially with a focus on retinal disease and treatment. So this session is being recorded. So if you or your colleagues would like to view this at a later time, you will be able to do so on the Foundation's YouTube page. Again, for those of you who have joined a little late, my name is Chad Jackson. I'm the Senior Director of the Preclinical Translational Research Program here at the Foundation Fighting Blindness. Just some housekeeping items. First and foremost, what's most important is asking questions. Dr. Rowe-Rendleman has developed a very robust slide presentation for us today, which will help formulate our discussion. However, if you had a question, please put it into the Q&A section, which should be at the bottom of the Zoom link here. Please try not to use the chat because in the Q&A box, we're able to record those questions. And if your question isn't answered during the session, we will be able to get to it at a later time. So please, again, use the Q&A box. But without further ado, and I don't want to take too much time up because we have a lot of information to get to, I'd like to introduce our speaker. Welcome you to the webinar. So Dr. Rowe-Rendleman, thank you so much for joining us. And if you wouldn't mind, could you just start us off by telling us a little bit about your professional background?
Sure, Chad, thank you. And I'm really honored and humbled that Foundation has asked me to do this, to sort of share what I have learned in ophthalmology and specifically in the regulatory field over the past 30 years that I've been in ophthalmology. My training has always been in ocular disease. I started at Princeton in ocular diseases, planting supernumerary eyes in frogs to see how they would hook up. But my postdoctoral fellowships were done both one in developmental diseases of the eye at the University of Texas and degenerative diseases of the eye at the University of California. And after that, I led a couple of programs in industry, particularly the macular degeneration program at Geron Corporation, the retinal disease program at Insight Vision in Alameda, California. And I also was in charge of the 3 glaucoma drugs medical education at the largest med ed company in the world, Sudler and Hennessy. One of the things I learned during all that time was that a lot of the work that we do in ophthalmology at the bench never gets to the bedside. And I wanted to know why. And one of the reasons I found out was that many small companies don't have ophthalmic expertise there when they go to the FDA. So they're kind of shooting in the dark. And since this ophthalmology is such a small niche, and I had a lion's share of the niche, I decided to start Omar Consulting Group 22 years ago. Omar Consulting Group is in Princeton, New Jersey, and Durham, North Carolina. We have clients in the US and in the EU and in Japan, and we help take people to the FDA, particularly those in the ophthalmic space. So I'm really happy to be here.
Well, we thank you again for being here with us, and thank you for sharing a bit of your background with the group and with the foundation. And this topic is so important to us because, as you know, it aligns directly with our mission in getting treatments and cures to the patients that need them. So please, let's just go ahead and go to the next slide, I think, and let's get the session started. So there's a number of questions that we're going to sort of structure our conversation around um, to help drive the discussion today. Again, if you have a question, please put it into the Q&A box at the bottom of the virtual page, not in the chat, in the Q&A box. So the first question is, can you please give us an overview of each agency's role, structure, and key functions? And the topics that we'll cover will be primary responsibilities of FDA and EMA. How do the roles of the FDA and EMA differ? Key organizational structures of the FDA and EMA, and common challenges drug developers face when navigating both the FDA and the EMA.
Okay, thanks, Chad. Thanks for kicking it off. And let's start with the first slide. I created a slide to sort of establish what were the functions of the different agencies, FDA and EMA. And both agencies have something in common, and that's to bring high quality and efficacious drugs to the public of each of their respective territories, the US for the FDA and Europe for the EMA. FDA regulates more than just drugs. It regulates medical devices, it regulates cosmetics and most foods, and it operates under the Federal Food, Drug, and Cosmetics Act. The EMA, which was established only in 1995, regulates human and veterinary medicines, and it does pharmacovigilance and under several different regulations. As I said before, the territories for the FDA is the US states and our territories. And also any foreign actor who happens to want to do business in the United States, the FDA regulates that as well. Whereas in the EU, they regulate 30 countries, and including Norway, Iceland, and Liechtenstein. When we talk about the organizations for these, we think of the FDA, the drug part of the FDA anyway, as three divisions that we all work with. One is CDER, which is the Center for Drug Evaluation. Then there's CBER, which is the Center for Biologics Evaluation. And there's a CDRH, and that's the Center for Device and Radiologic Devices. When we look at the EMA, we work with 6 scientific committees, which are populated by representatives from each of the nation states, plus members of the EMA staff. And those committees include, and I won't go through all of these, but for our important purposes, the CHMP is the Committee on Human Medicines. So it's really different in how these two agencies are set up, but the goal of the two agencies is quite the same. Now, there are a few differences in what these agencies do, and pardon this very busy slide, but there are 2 things I wanted to bring out. First of all, risk evaluation is managed differently by the 2 agencies. In the FDA, the risk evaluation is done by drug. So the FDA identifies a particular drug that requires what we call a REMS strategy. That's a risk evaluation and mitigation strategy. And it applies only to those drugs that have serious safety concerns that have been identified. And once a REMS has been applied, it doesn't vary across states as it does in the EU. And the FDA is the central body for determining whether or not there's going to be a REMS strategy. On the other hand, the risk management plan is for all medicinal products. So that's in place at the very beginning. The main components of the EU risk management plan is safety specification, pharmacovigilance, and of course, risk minimization. Because the EU is made of so many member states and those populations differ quite a bit, the national authorities in the states can adjust the risk management plan to align with that particular member state. So those are two differences. That's one difference. And the second difference that I wanted to state is about reimbursement. The EMA is involved in reimbursement of drugs, whereas the FDA is not. The FDA leaves that job to our CMS, which is our Center for Social Security and Medicare. So moving on to your third question, Chad, about common challenges that drug developers face when navigating both the FDA and EMA. This slide is based on my experience and only my experience. I'm sure others will have different types of challenges that they've come across. But in the 22 years that I've been doing this, the common challenges have usually been regulatory hurdles where folks aren't understanding the endpoints that demonstrate safety and efficacy, and that these endpoints could differ across countries and regions. So it's really important to consult the regulatory agency in meetings, these early meetings, pre-IND meetings about endpoints, but don't depend on the trial registries such as clinicaltrials.gov, because the agencies don't regulate clinicaltrials.gov. So the endpoints that are in those clinical trials may not be endpoints that the FDA accepts. Development timelines and costs is another challenge, because most folks underestimate those development timelines and costs. It's very expensive and time consuming to develop a drug. So it's important that sponsors, folks who are looking to develop new drugs, surround themselves with people who can help them understand the variables that go into timelines and the cost of developing a drug before making timelines, and definitely before speaking to the public or speaking to an investor. Clinical trial challenges. The recruitment of populations and the preferred medical practices definitely differ across countries and regions. So an acceptable clinical trial that we all envision or have seen others do may take a long time to enroll in the United States. So we have to check with the CROs first to understand what are their assumptions when they hear a particular disease or indication that we wish to create a clinical trial for. Then we can design around those assumptions or with those assumptions so the trial can be enrolled in a timely way. Finally, patient use and access. We have an obligation in our clinical trials to demonstrate cost benefit. And if we don't, it can prevent the study from moving forward. And I don't mean at the FDA, but you may not even get funding for a study if you can't demonstrate this cost benefit for patients.
We do have a quick question. I think it's just before we move on, maybe two questions here. One, I believe this is just sort of a question for confirmation. Is that the question states, or the statement states, I understand that pricing and reimbursement in the EU is determined on a country-by-country basis and not by the EMA. Can you confirm that statement? Sure.
Yes, the countries, the national authorities in each of the different nation states in EU do contribute to the pricing, and they will discuss with the EMA what pricing is best for them. Each of them will roll out a drug at a particular price, and the EMA has already set sort of a guideline for pricing based on those discussions that they've had. So much of the pricing is done by committee, and the nation states will select the prices based on what the committee decides the range will be. Is
All
that
right. Yeah,
so?
I think you've answered that. Yeah, we can move forward now. Thank you. And also, I'd like to encourage the group again. Please do not use the chat to ask questions. Please enter your questions into the Q&A box. Our next question is regulatory pathway for drug and device approval. The topics covered in this section will be high-level overview of the regulatory pathways for drugs. Do the regulatory pathways differ for new drugs versus generic drugs, biosimilars, or biologics? What are the key differences in the FDA's new drug application process versus the EMA's marketing authorization application and expedited programs that are available through the FDA and EMA?
Okay. All right, I wanted to start off then, as you suggested, with a high-level overview of the regulatory pathways and drugs. And this high-level overview is sort of to give you a lay of the land when you are developing a drug, because it takes a long time, and there are certain parts that you shouldn't avoid. In the very beginning, and most of you are familiar with, is doing basic research at the bench. And that basic research could lead you to early discovery. And then you'll do early discovery proof of concept studies. And from there on, you'll move on to a different type of basic research, which is called GLP, or Good Laboratory Practices basic research. And this is the IND enabling research that you use to get you to the IND application. But all while that's going on, there's another set of research going on here down in gray. You're developing your formulation. Most of us work with a pre-formulation when we're at the bench and doing basic research, and even during early discovery. It's proof of concept. But during the basic research GLP phase, you've got to figure out how to scale up and make a real formulation for the eye. And that takes about 1 to 6 years to do all of that work. And for some people, it's taken much longer. But your IND application has to include your basic research, your early discovery, and your GLP studies, in addition to your scale-up formulation. Not only does it need a scaled-up formulation, but it's important before you go into clinical trials that you have completed GMP manufacturing of at least an engineering batch. That's a batch of your drug that fits all of the criteria that you set for it and is everything but GMP. Then you can continue with the beginning of your first trials, perhaps your phase 1 trial. And you'll continue creating and developing your formulation. Perhaps you'll put it on stability, and the stability studies will go on during the time of the clinical development. Also, you might even change your formulation a little bit, and don't be afraid if that has to happen. And that's just an amendment that's going to be submitted to your IND if a formulation is changed. It's also submitted to your annual report if your formulation has changed, because you might find some things during your phase 1 or phase 2 studies that suggest that a formulation change is needed. So don't feel it that's a problem. As you move on to phase 3, though, you need to be in your commercial formulation. And the range of time for working through the human testing is about 10 years for some. So the first phase is probably the most important. As everyone knows, the foundation is the most important thing. So be sure to design your phase 1 study such that you get the information you need to make decisions about formulation or the population that's going to be used in phase 2 or the size of phase 2. After that, we move on to the NDA or the BLA application, depending on whether, respectively, you're going to be submitting for a drug or a biologic. And after that, the FDA begins its review. And the review can take up to a year. So give yourself time. And after review, depending on your drug, there might be some post-market monitoring or some post-market requirements that the FDA suggests to you. And those requirements, depending on your drug, could go up to 5 years. And with some biologics, especially some vectors, vector viruses, it's 5 years for safety. So that's going to be important to remember when you are putting together at a high level what your clinical program will look like. Moving on. So what are the types of drug applications and regulatory pathways? Once again, we're starting at the basics, and that is starting with the IND. And everyone who intends to commercialize a drug needs to submit an investigational new drug. If drugs are used across state lines, you need to have this IND, which is an exemption to you to allow you to run a study in which drugs or parts of your drugs are moved across state lines. That means even if you get bottles from someplace else to put your drug into, you will need an IND. Alternatively, there's something called a research investigational new drug or a research IND. But in this case, you are going to promise the FDA that you're not going to commercialize any of the work that you do. And generally, this is held by an individual investigator or an academic institution or a nonprofit entity. The bar for a research investigational new drug is very low. You still should meet with the agency to talk about what you plan to do. But the paperwork for such a thing is usually very, very low. If you have the new drug, the capstone is an NDA. That's a new drug application. And there are 2 paths that your new drug application can go. I think you may have heard of the phrase 505(b)(1), which is for a new drug, and 505(b)(2), which is for a repurposed drug, a drug that has been used perhaps in another indication, and now you're repurposing it to be used in the eye. Same thing goes for biologics. The biologics applications, they have a path that's called 351A. That's for a new biologic. Then they have a path called 351K. That's for a biosimilar. And then, of course, there are generics. And generics are drugs that are generally drugs that are created from other drugs that have already been approved, may now be off patent. And so the type of NDA that you need to submit is called an abbreviated new drug application. It's abbreviated because the clinical and animal studies are generally not required to get the ANDA. And that path is called a 505J.
Can we take just a quick pause here? There is a question here I'd like to get in from one of our attendees, and that is, and this speaks to something that you sort of touched on a bit earlier in the previous section, but is there a difference in the way that FDA, CBER, and CDER divisions operate as compared to CHMP and COMP in the EMA regarding rare disease?
Both agencies have rare disease divisions or orphan disease divisions. If you are going to be going to orphan disease in Europe, you would go through that division. In the United States, however, if you have a rare or orphan disease, you will still go to the same centralized body, either CDER or CBER. You would let them know on your initial documents that you are dealing with a rare or an orphan disease. And when you submit your protocols, there can be special exceptions or exemptions to you because you're dealing with a rare or orphan disease population. If you're a European citizen, you might also be able to take advantage of some of the cash incentives for studying rare and orphan diseases in Europe. And at the FDA, it's more of a consideration for more meetings with the FDA instead of money. So those are some of the big differences for dealing with the two types of diseases in Europe and United States.
Right. I think we probably can move forward. There are other questions, but I think that you'll touch on some of these questions later on in the presentation.
Okay. So I spoke about generics at the end of the last slide, and I wanted to sort of capitalize and let you guys know, what do people think a generic is? We always think of it might be the brand name, but maybe not as good. But there are some rules about generics, and those rules include that the active ingredient has to be the same, but it can differ in terms of inactive ingredients such as dyes, flavoring agents, and binding agents. The dosage strength and the dosage form has to be the same as the parent drug, but it can differ in things like size, color, and shape. The route of administration for a generic and its performance in the body, and that's very important, the performance in the body, all have to be the same. And it's based on bioequivalence studies. But bioequivalence studies doesn't mean that it has to be 100% match for match for what we call the reference listed drug. There's about a 20% wiggle room there for performance in the body. So that is one of the reasons why some folks think generics aren't as good as brand name, but that 20% may not mean anything to a person who takes a drug chronically.
Another quick question for you before we move on. Sorry, keep interjecting here. But in the 2 slides ago, when we were talking about the drug applications and the regulatory pathways, Dr. Shigemi Matsuyama would like to know, can you first start with the research investigational new drug process and then move to the IND process?
Only if it's a completely different drug. You will make a statement to the FDA that for your research IND, you will not commercialize that particular drug. However, let's say there's a family or a class of drugs that you want to study, and you're not sure if you want to make that into an IND right away. So you just want to know if a class effect occurs. So you can study a drug in a particular class, promise not to commercialize it because you won't. And then you can go on and use your own facilities to do some design of a different drug based on that class, because now you know the class effect. And that will be a new chemical entity. And you can get the regular IND for the new chemical entity.
Thank you.
Okay. So this slide that we've had up for a bit, it's given a lot of people a chance to take a look at it, is how do new drugs differ from generic drugs? And we've talked about the non-clinical part. We've talked about the CMC part, but let's talk a little, and we've also talked about the bioequivalence section. But let's talk a little bit about the top line here, and that's exclusivity. And as I stated, a generic comes on the market after a brand name typically has lost its patent or the patent has expired. So those brand named drugs, they have exclusivity, meaning there can be no pressure from a generic while they're within that exclusivity phase. However, once a generic comes on board, then there is little exclusivity left, and generics alone have no exclusivity. So the FDA is the only agency that allows exclusivity, and that's one of the things that one should discuss when you go to the FDA about exclusivity. And there are ways to extend the exclusivity beyond the patent life. That's not for this discussion here, but this is work for your regulatory lawyer. will tell you about ways to extend beyond the exclusivity afforded by your patent. Let's move on to how do biosimilars differ from generics. Biosimilars are usually made from living sources because they are biologics, whereas generics are generally made from chemicals. Those are drugs. With a biosimilar, they require a very specialized process. The process that they have may not be the same as the process of the original biologic. But in generics, they're actually encouraged to use the same process that the innovator used. Their biosimilars are very similar to the biologic, but they're not identical. As a matter of fact, because they are biosimilars, they're the entire idea of that type of drug is that it's not identical to the original. But generics are original, and the original are very, 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 very close. They are basically a copy, plus or minus 20% of the brand name. Each of them enjoys faster development because they can use public information that was not available to the innovator. And each of them is usually less expensive than the innovator.
But before we move on, we have another quick question around exclusivity. So this is from Mark Panisi would like to know, how strong is exclusivity protection for rare diseases? If a competing treatment comes along, do they need to show superiority to break exclusivity?
Yes.
Simple enough, huh?
Absolutely.
Thank you.
So now I'm going to talk a little bit about the biologic versus the biosimilar. We've already stated the issues about patents. Biosimilars are not patented, whereas biologics are. They have a different regulatory path, 351A for a biologic, 351K. Are they identical? Absolutely not. But they should have, and they must have the same mechanism of action as the reference product, but only to the extent that the mechanisms are known. So if a new mechanism, for instance, of how Lucentis works becomes available, for instance, then a biosimilar can take advantage of that new mechanism and perhaps even become what's considered a new biologic. But that's really a regulatory question. As far as non-clinical toxicology, that's absolutely required. Pharmacology, pharmacokinetics, and toxicology must show comparability, and the biosimilar has to show comparability to the original biologic. And yes, they all have to run safety trials and efficacy trials. So part of this conversation was to kind of talk about the differences between FDA and EMA and how that NDA and the MAA work. I'm going to start with step one, and that's the IND versus the CTA. So the clinical trial authorization versus the investigator's new drug exemption form. In the US at the FDA, we file a single IND and we file it per program. In Europe, of course, the clinical trial authorization is filed per trial, and you can file for multiple member states at the same time. In the United States, the IND is a huge document. It's composed of 5 parts. We call them Module 1 through Module 5, where Module 1 is administrative. Module 2 is a summary. Module 3 is the chemistry, manufacturing, and controls. Module 4 is the non-clinical studies and study reports, and Module 5 is clinical. You have the same thing in Europe, but you also have to submit a dossier to the ethics committee at the same time, and both are reviewed at the same time. As far as review time is concerned, review time is 30 days for the FDA and 45 to 50 days for the EU. And during the review time, both agencies can ask you for more information. The ethics committee in the EU can ask you for input, and there might be some issues with the way you validate your reasons for doing things a certain way. The FDA can put you on clinical hold, and so can the EU. And a clinical hold simply means that somewhere in your dossier, there were unanswered questions about either your manufacturing or your clinical study. Rarely does it have to do with anything about your non-clinical studies. If there are no questions from the FDA, you will not receive any information after the 30 days. The 30 days elapses and you're free to go. However, if there were questions from the FDA and a clinical hold was installed, you will receive a safe to proceed letter. In the EU, you get a notice, a safe to proceed letter of whether or not your clinical trial was acceptable with modifications, not acceptable, or refused. You will get that letter no matter what. And it's up to you to notify the EU whether you're going to start, withdraw, or halt your clinical trial. In the US, with an IND, whenever you make changes or whenever you go to another trial and you're going to start a new one under the same program, you add an amendment to your IND. In the EU, you submit a new CTA. So slightly different, but the EU tries to work with and gives you more times to meet with them, although you do pay a fee for that, so that you can, this CTA submission is painless. Okay, I'm going to take back that word. It's not painless. It's just, you feel a little better. Finally, what about programs to expedite drug development? And I'm going to focus on the FDA because that's where I work the most. I'm not very familiar with those that happen in the EU. But in the US, we have 4 programs. First program at the very beginning, a program that doesn't require any clinical data, is the fast track designation. But in order to get a fast track designation, your drug must fill an unmet need, and there must not be another drug on market that fills that need. So this is once again where you have to go back and look at indications, and if there's a drug that has an indication similar to yours, and it's doing that job, you won't get fast track designation unless you can demonstrate that your drug is for a different population and is filling an unmet need in that population. So what do you get? You get the ability to have rolling submissions, meaning that you don't have to submit all parts of the IND at one time. You can submit them as you get them. And then you have additional meetings with the FDA all along. So for some, that's a great thing. And every time a company gets a fast track designation, you'll see them talking about it on their websites because this allows them to have more FDA input onto every step that they do. The next is the breakthrough therapy designation, which is different from the breakthrough designation because the breakthrough designation without the word therapy refers to devices. But now we're talking about drugs. So the breakthrough therapy designation is a designation that you can apply for. You can apply for with your clinical evidence. And the best time to do it is no later than the end of phase 2 meeting. And to do it, you request a meeting with the FDA and you present your argument, just as one does with the fast track designation, for why you believe breakthrough therapy designation should be given. Your clinical evidence has to suggest substantial improvement over existing therapies. And the wiggle word there is substantial. And that's up to the FDA to review and for you to convince them. What do you get out of this? You get prioritized review, you get intensive guidance, just as you get with the fast track, and you also get the ability to have fast track if you want. So that's the additional, that's the rolling submissions piece. Third is accelerated approval. An accelerated approval is something you apply for at the time of your NDA. When you're submitting an NDA, you've had numerous meetings with the FDA. You want to decrease the FDA's time from 12 months to 6 months. And typically, accelerated approval is reserved for serious conditions that are based on surrogate endpoints. We have a lot of surrogate endpoints in retina. So we have very few drugs, though, that have received accelerated approval. Once accelerated approval has been given, it's an approval, but the sponsor will have some additional responsibilities after approval. And that's important because if you don't carry out those responsibilities, and if the results of those clinical studies that are part of that responsibility are not as they were expected to be, your approval can be pulled back. And then finally, there's priority review. This is something that the FDA designates. We don't designate it for ourselves. And they typically designate a priority review at the NDA time. And once again, this gives you the 6-month review time. Okay, any questions for that section?
No questions for that section. I believe we'll probably be able to handle them at the end. We are running just a little bit behind time with our discussion, and we have just under 20 minutes left, so we might have to pick up the pace just a bit. But for the audience, I still want to encourage you to please put your questions into the Q&A box down at the end, at the bottom of the virtual page, because if we're unable to get your questions answered now, we will definitely follow up with you after the meeting's over. So please, I would like to encourage you to continue to send in those questions. But our third discussion point revolves around regulatory engagement meetings. And here we'll talk about the description of the FDA Interact and pre-IND meetings and preparation and discussion during these meetings and the level of feedback that you can expect after you have one of these meetings, and maybe guidance documents that can help guide you a little bit during your journey with these types of engagement meetings.
Okay. So this is about Interact, which means initial targeted engagement for regulatory advice on CDER and CBER products, and pre-IND meetings. Interact meetings are typically early meetings for novel products, and they are limited in their agenda to CMC and non-clinical results. And the responses that you get from the FDA are non-binding. Pre-IND meetings, on the other hand, are intended to discuss your full IND plan prior to submission, and the results are binding, but the FDA is not rigid, so you could change your mind at the IND time if you supply justification. The timelines for these meetings is typically 60 days. There's one big difference. The pre-IND meeting, you have time after you have requested the meeting to submit a meeting packet. But during the Interact meeting, there is no time. You must submit your meeting packet at the time of the request. So remember that. You don't want to be the type of person who has been denied because they didn't submit their meeting packet at the time of their request for Interact. Some pearls for preparation and discussion is basically remember your timing here. When you submit your request letter, have your preliminary questions there and background information. If it's your Interact meeting, the background information is important. In the pre-IND meeting, you can have a very abbreviated background section. Prepare before you submit your request letter. Make your pre-IND package, because there really is only 9 days left. I mean, once you get I'm sorry, only 30 days left. Once you get your meeting granted letter at day 21, you've got to get your request package in 30 days before the meeting. That's not a lot of time to work on a package that's got that much information in it. Here, the slides will be available to everyone. The level of feedback is based on the type of questions you ask. If you ask questions that are kind of nebulous and not very clear, then you're not going to get a very clear answer. In your pre-IND meetings, you will include information on your product or your device. You'll include all of the pharmacology information that you have. You will not include your GLP studies because that's for the FDA to help you design. And you'll include a clinical synopsis. So you'll get these questions if you request the slide deck after this meeting. So there are some questions that are just not appropriate at any of these meetings, and those are jurisdiction questions and questions that are related to other people's product. So you can't ask about that because the FDA won't tell you. Okay.
All right, we do have some questions sort of in the intermediate here that I believe we can take. I'm going to take these a little bit out of order. From Shigemi Matsuyama, we have question is, are rare disease indications, do they automatically have accelerated reviews? Is that correct? And then what is the difference from other fast track review?
The question is whether or not an accelerated review automatically kicks in for a rare disease.
I believe so. I believe that's
I'm not sure if that's true.
Okay. All right. And maybe this is a little bit too early, but I'll ask the question anyway. This one is from Sandeep Chandra Bolapalli. Could you shed some light on the regulatory pathway for AI or machine learning based models for disease screening or protection?
A little bit outside the scope here, and I cannot.
Yeah. Okay, fair enough. And I believe we have 2 more questions here, but I think we can hold on to these until the end.
Okay. So we're ready for question number 4 about the submission.
Sorry about that. I got distracted with the questions. Again, so the last point of our discussion is question 4. Submit is about the submission processes. A step-by-step guide on preparing and submitting applications, including some pitfalls and how to avoid them. The particular things discussed in this section will be components of a successful FDA, NDA, or EMA, MMA submission, MAA submission, not MMA. We're not here to fight. Meeting all regulatory requirements and guidelines, common pitfalls in preparing and submitting drug approval applications, a walk through the typical review process for an FDA and EMA drug application, and an effective communications and strategies.
Okay. All right, so let's take a little walkthrough of a successful EMA, MAA submission. These submissions should start 18 months before you intend to actually submit. And the European agency allows you time to have what are called pre-submission meetings. So first off, after you've completed your phase 3 studies, you need an eligibility request. This is a meeting with the agencies to determine if you're even eligible to submit an NDA or an MAA. Then you notify them after that meeting of your intent to submit. Then they will appoint reviewers. These are called rapporteurs in EU. After that, rapporteurs might have some questions for your submission. So these are pre-submission meetings. And then after you've heard from the pre-submission meeting, there might be some changes you need to make to your dossier. So your submission date may change. So you need to reconfirm your submission date with the agency. And then you make a submission. So there are lots of times, at least One, eligibility, pre-submission, where you are able to speak with the agency and should speak with the agencies about your MAA to make it successful. But even with that, there still might be some issues at the end of your submission. On the other hand, let's look at what happens at the FDA. About 12 months out, once again, you'll have a review meeting with the FDA. This is usually your end of phase 3 meeting, your pre-NDA meeting, and then you submit an NDA application. It's important that you ask as many questions as you possibly can. You get the FDA to agree on the endpoints you use, the analysis that you use, and any other things that might be part of your NDA application. The FDA will decide in 60 days whether or not your NDA application should be accepted. Then they'll assign a review team, just like the Europeans do with the rapporteurs. Then for the next 6 to 7 months, your NDA will be reviewed. The FDA may also want to do some inspections of the facilities you used. If your facilities are not ready for inspection, this could hold up the approval. If there is a brand new drug, the deputy director or director of your division might ask for an advisory committee, in which time you will come before the FDA and you will defend what's in your NDA. And after that advisory committee meeting, your drug will be approved or a response letter will be given to you. A response letter means your drug was not approved. So here's a graphic of this whole thing that I just mentioned. The EMA at the top, marketing authorization, taking just a little under a year. But remember, there are times here where questions have to be asked and the clock stops. So sometimes we might say that it takes a little longer than a year while this clock is stopped, while you're talking to the rapporteurs and answering questions. In the United States, we have a couple of things to think about here. We go ahead and submit after we've had our discussions with the FDA. The FDA may have some follow-up questions for us, but we do have rules in the United States about how long it takes them to respond to your NDA, and that's usually 12 months. If there's a priority review, that could be 8 months, but a standard review is 12 to 14 months. So don't be surprised by that. What are some of the pitfalls? The whole point of developing these drugs is to balance the risk with the benefits. So some of the common pitfalls in a very mature clinical program is a failure to demonstrate efficacy, as efficacy is designed or somehow explained to you by the agency. If you still differ with the agency about efficacy, there's still a route for you to take, but it won't be at the approval of your NDA. Sometimes when looking at an NDA, the FDA may decide that additional studies are needed. And by additional studies, they may look at your study and say, you need more people. We now have a mandate in the United States to try to include both genders, if possible, and all ethnicities, if possible. And then different types of people. So your study population may have been skewed to one side, even though the discussion with the FDA was to broaden your study population. So those are things to think about. And finally, there might have been insufficient exposure time for your study. And some sponsors who might decide that instead of doing an 18-month study, that a 12-month study is sufficient. And that's insufficient exposure time, according to the FDA, for some drugs. And therefore, your drug won't get approved. Some of the bumps in the road are unexpected safety issues. This happens to anybody, especially in phase 3, where your study population is broader. There may be manufacturing issues at the plant that you chose. So that's not your fault. That's a bump in the road, but you'll have to find an alternate manufacturer. There might be an undependable drug supply. The FDA is responsible for making sure that once your drug is approved, it's going to be available. And if your drug supply is not dependable, your drug won't get approved. And if your facility is not ready for inspection, that inspection is really important. Tells the FDA whether or not your drug quality is going to be at the level that you say it is in your NDA. So sometimes these inspections are surprise inspections. So your facility has got to be ready. Finally, let's take a walk around how the drug review process works. We started off this conversation with preclinical animal testing, and it's strongly suggested that you have a pre-IND meeting. And now we know you can have both an interact and a pre-IND meeting. And from that pre-IND meeting, you will move on to your GMP manufacturing and your GLP studies. That information will go into your IND, and that will allow you to start your first studies. It could be a phase 1/2 for diseases of the eye, because we often start our studies in patients. And after that study, it's important to have a conversation at the end of phase 2 meeting about what the endpoints are needed for phase 3, or what additional phase 2 studies might be needed. After the phase 3 study, please go to your pre-NDA meeting, and then use the pre-NDA meeting to get all the information you need to submit your NDA, and get ready for your next drug. Start your preclinical testing all over again. So with that, I want to thank everybody for staying on. I think we have about 4 minutes left for questions. This is my contact if anybody needs it. I think we will make the slides available to you all, as well as the responses to the questions, especially questions I didn't know the answer to. I'll look them up and get them to you. Chad?
Well, thank you so much for that detailed presentation and discussion. We do have a few questions, so let's just jump right into those to see if we can't get through some of them before our time runs out. So the question I've been holding on, I think it was actually probably one of the first questions, is what about efficacy endpoints in retinal degeneration or retinal degenerative diseases? Does the FDA respond to this or to your questions on this at the pre-IND stage?
Yeah, it's really important to get the FDA's buy-in at the pre-IND stage about efficacy. They will want to be convinced of an efficacy endpoint that you have. And often we propose efficacy endpoints that are not yet validated by our own professional group. So someone may have used them somewhere, but they aren't validated for the population. They aren't validated by our own, by all the ophthalmologists. So that's one of the sticking points for the FDA, is to use these validated endpoints. So often they will want you either to do the validation work yourself, or to come back to them with some kind of consortium, which says, we all agree, and these are our experiences, and this is how it benefits the patient, and that helps to validate that endpoint.
Having about 2 minutes left, we have a question here from Claire Gelfman. It says, can you speak to the role of animal efficacy models in the regulatory process for rare and not so rare retinal diseases?
Sure. First of all, let's go back to that word validation and animal models. Animal models are animals. They don't really replicate the same process in humans. So we use a model, maybe several models, to try and demonstrate 1 or 2 or 3 different points. The FDA is basically model agnostic. They want you to provide the justification for the model that you're using and the drug and the pathway that it's supposed to take. So there's, it's not as if the FDA has a list of expected models. That doesn't happen. As a matter of fact, if you go there and you ask that question, which I have, you usually get a pushback saying, we don't really want to tell you about any models, but if you have a new model, then you justify it to us on paper and with experiments as to why this model is representative of how your drug behaves.
All right, thank you. And maybe we'll end off with this question here. I think it's a good one. It says, I think for you personally, what regulatory challenges have you faced that are specific to retinal diseases?
Specific to retinal diseases is often delivery, the delivery of drugs to the back of the eye. We now face this issue of how we're going to get drugs to the back of the eye, whether or not we're going to use a new device or whether or not we're going to have a combination drug and device regulatory pathway. And that pathway is not very clear to us or the FDA or the Division of Combination Drug Therapy. So this is probably the biggest hurdle is understanding what is going to be the delivery of the drug to the back of the eye. If you're going to use something like intravitreal injection, which has already been used many, 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 many times, that path is fairly simple, but novel pathways of trying to get something to the back of the eye, minimally invasive, that becomes a little bit more difficult.
Well, we are out of time. Thank you again so much for this presentation, as it was so informative. We have many kudos to you, Dr. Rowe-Rendleman, in the chat, so I'll let you read those. But I'd like to thank our audience for attending. Again, this is being recorded, so it will be hosted on the Foundation's YouTube page at a later date. Then we also will answer your questions and make the slides available in maybe like a week or so. So stay tuned for that as well. If you have any questions in the meantime, please feel free to reach out to me, and I will get your questions answered as soon as possible. Thank you all for joining, and that concludes our webinar for today. Thank you.
Right.